For millions of years, Mother Nature has nurtured all creatures great and small, protecting them from strife with the tranquil, slow elimination of harmful substances from land and seas. While EcoImpact just cleans it!
The Rhodes Peninsula is an industrial wasteland. Over the last 2 centuries, it has hosted a series of environmentally disastrous activities which have left the soil soaked with pollutants and the water saturated with poison. In the 18th century, the peninsula was a residential area. In 1930, Allied Feeds established a flour mill on the site. A timber treatment plant was added in 1942, then a gasworks. Then during World War II, an explosives factory. Union Carbide reclaimed land using industrial waste, then pesticides and herbicides were made on the site. Chlorobenzenes, chlorophenols, dioxin, furanes, and Agent Orange. Today, internationally renowned environmental scientists agree the peninsula is one of the most heavily polluted sites in the world.
Current remediation by developers has pushed down the levels of most pollutants detrimental to the environment or to human health. But, according to the National Environmental Protection Schedule, the levels of chlorobenzenes, PAH, and dioxins still exceed the HILD standard and need immediate attention. In order to meet the HILD standard, we must lower the levels of these pollutants to 1% of their current levels. The traditional narrow-minded way of approaching this problem is to use a messy technique like thermal desorption, or a slow, tedious technique like in situ biodegradation. EcoImpact knows that developers can't wait around. Our method uses cutting-edge technology to bring the entire site to standard within 1 year at a competitive price. Let's take a look.
First, the site on which the plant is to be constructed will be excavated to a depth of 5 meters. This allows construction of the plant to commence immediately on the floor of the excavated pit, the walls of which will be reinforced with concrete. Once the plant is constructed, excavation of the rest of the soil will commence. The Rhodes site will be partitioned into 10 meter by 10 meter cells. Excavation will begin at the northmost cell and work southwards in east to west rows. This prevents rainfall from washing contaminants from untreated soil into the cells which have been backfilled with treated soil. Water sprays will be used to suppress odor and dust emissions. Excavated soil will be transported by truck to the stockpile area of the plant. Once the soil has been treated, it is backfilled in the same east-west pattern. The Homebush Bay sediments will be extracted using a floating dredger, which pumps the sand into a waiting truck. It is then processed in the same way as the soil. Silt curtains will be put in place to prevent sediments from building up in unwanted areas in the bay.
Now onto the plant itself. The lower part of the plant machinery sits at the base of a pit, 5 meters below ground level, while the upper part is contained within a warehouse. Let's take a bird's-eye view of the path contaminated soil takes through the plant. When a truck carrying excavated soil arrives at the plant, it dumps it through the warehouse dock. Water is sprayed onto the soil to minimize dust pollution. A soil grader separates out large rocks and waste, dropping the soil into this covered stockpile area. At night, when excavation ceases, the dock can be closed to add an extra layer of soundproofing. The majority of the plant is underground, and as this graphic shows, the upper portion is well sealed and whisper quiet. When an extraction tank is ready for filling, soil moves up one of these conveyor belts, where it falls into the tank along with a stream of preheated water. There are 9 of these tanks, each of which can be filled and emptied separately. Each tank is clad with thick thermal insulation.
Let's pause for a moment to look at the cutting-edge technology behind the extraction tanks. For decades, chemists have known that water above a critical point of 374 degrees Celsius is capable of dissolving certain organic compounds which at normal temperatures are insoluble. Unfortunately, water at this temperature is corrosive and hard to manage, and hence impractical for soil treatment. However, recent research has found that certain combinations of high pressure and temperatures below the critical point can also dissolve specific organic compounds. Both lab and pilot-scale studies have proven that subcritical water extraction can extract all the pollutants EcoImpact is targeting from nearly any kind of soil. In approximately 80 minutes, our extraction tanks literally wash the soil clean of chlorobenzenes, PAHs, and dioxins. As an added bonus, the high temperatures reduce some of the dioxins to harmless aliphatic hydrocarbons.
Back to the plant. Once filled, the top of the extraction tank is closed with a hydraulic hatch. Pipes connect the top and base of each tank into a circuit of water, which is kept continuously moving by this pump. When a tank is sealed, the valves at each end open and water begins moving through. The pump and the heater on the main water circuit cycle the water through a series of temperature changes, which maximizes the efficiency of extraction. The pressure is kept constant. This chromatograph monitors the dissolved pollutant content of the output water, giving feedback to a control computer, which can recalibrate the temperature changes to target particular pollutants. Once the chromatograph reports that the tank is free of pollutant, the tank is disconnected from the circuit, and a hatch at the bottom dumps the soil onto this conveyor belt. The belt carries the soil to a belt press, which removes residual water for recycling, and then dumps it directly into a truck which backfills it into the site. The contaminant water output from the extraction tanks is fed into the centrifugation vessel and mixed with powdered activated carbon. The activated carbon binds up all pollutants found in the slurry. The contaminant-bound activated carbon is then separated from the liquid phase via vortex centrifugation. The vortex centrifuge is a patented process for rapidly siphoning suspended particles out of a liquid. An electrical motor induces the water into a vortex flow. The centrifugal force drives the heavy contaminants to the vessel wall and into the collection tank in the vortex base. Lighter contaminants and gases are passed through the central outflow line and into the collection tank. The clean water passes through the annular outflow chamber to be recycled back into the extraction vessels. Once the collection tank is filled, the vortex is isolated from the circuit. The tank is removed and a fresh replacement brought in. The filled tank must be handled carefully, as it contains high concentrations of dioxin and other toxins. As soon as it is removed, it is inoculated with microbial starter culture and sealed with a specially adapted lid. Hoses for oxygen and nutrient injection are attached to ports in the lid, and an agitator is inserted. The tanks are then stacked in the storage area below the conveyor belts.
The microbial inoculants consist of 3 bacterial species: Sphingomonas yanoquiae B1, which degrades PAHs, Sphingomonas whittichi RW1, which degrades dioxins to chlorobenzenes, and Escherichia heromonii, which degrades chlorobenzenes. These species have been chosen because of their ability to degrade all 3 pollutants to below the HILD level within a short period of time. Based on past experimental results, the time required to fully remediate each tank of sludge will be 22 days. The nutritional requirements of the bacteria are simple and can be met with commercially available media. Oxygen and agitation within the tank will encourage rapid microbial growth and activity. To save energy, the bioreactors will maintain an optimum temperature of 29 degrees Celsius using the same heat exchange pipes as the extraction tanks. Because each microbe can utilize its target pollutant as sole carbon source, the entire process is self-limiting and self-contained. Once the remediation is complete and the pollutant level is confirmed by chromatography, the harmless and nutrient-rich sludge will be simply spread across the remediated portions of the